So hello everyone. I welcome you to our webinar today. My name is Gabriel, okay, and I'll be standing in for Cecilia. I'll be managing the webinar today. I'll also be at the end of checking the questions from the Q&A session. So I'll be fully in charge today. Cecilia is unavoidably absent. So you see my face every time, not like the last webinar where I and Cecilia were shuffling. So before we have our speakers, I would like to give you a rundown of things we do at PAR and how far we've come for this webinar series. Okay, so today we are going to be having David, Olayinka, Ishola, Megan Coffee. We are going to be having Margaret, Lubwama today. So they're going to introduce themselves for like 1 or 2 minutes before they start their presentation each. But before that, let me continue. So, what do we do at PAR Foundation? So our aim basically is to eradicate the spread of antibiotic resistance. And if you are aware, last week was World Antimicrobial Resistance Week, of which if you followed our page, especially our Twitter page, you see that there are lots of videos, flyers, a lot of content flying around to celebrate the week. And our major offering, our primary project is actually grants making. So we support research and educative initiatives that are focused on prevention of antibiotic resistance. And we started in 2017. So globally, we have more than 1,000 people subscribed to our newsletter who are passionate about AMR because we have a lot of opening rate for our newsletters. And then we have funded 25 projects in Europe, Africa, Asia. And for the first time this year, we have a project that is funded from Latin America. And so far, we have funded with 16 million SEK. Up to this year, we funded projects with up to 16 million SEK SEK because we are Swedish. So our main currency is SEK. The way we communicate it is in SEK. So last week we had our World Antimicrobial Resistance week. So we are still trying to pull up our content for that because we're still trying to reach out to the winners so that we can give them their reward. So the first winner, we received 300 USD. David, can you mute yourself, please? Sorry, for a while, you can just mute yourself. So as I said, so last week we had organizations, two organizations who were first one, they were involved in converting and translating, a document on antimicrobial resistance into various languages. So they won our World Antimicrobial Resistance Week challenge. So they will have 300 USD. We have two other people who are runners up, who are going to get 100 USD. So you can just follow our Twitter so you get abreast with this information. So we have just 500 USD for our mini grant, but our major grant will be opening very soon again. So currently we have like 8 grantees for this year, which of course, if you follow our page too, you will definitely be able to get full information about our current grantees. So the first session is with David Olayinka Ishola. So he's a passionate veterinarian, I beg your pardon, from Nigeria, and is dedicated to animal health and welfare. So we'll be having nuances, of One Health in his presentation, and he'll be taking us through for the next 12 minutes. And after that, we are going to be having our Q&A session. So if you have questions at any point in time, you can drop it in the Q&A box or in the chat section, whichever you are able to meet based on your status on this webinar. Either you're a panelist or a speaker or just an attendee, you're just an attendee, maybe just get the link somewhere. Whatever access you have to communicate with us, either through chat or Q&A, you can send us your questions. So David, are you able to start now?
Yes, can you hear me?
Yes, please. So you can share now, unmute yourself, and if you want to share your video, you can go ahead. So guys, we'll be having David now. Your time starts now, David. Go ahead.
All right. Good afternoon, everyone. As it has been said earlier by Gabriel, my name is David. I'm a veterinarian with over 6 years of experience working in the poultry industry. Today, I'm excited to discuss the crucial topic of antimicrobial resistance prevention, drawing on my 6 years of experience in this field. And I discovered that while my presentation is a little bit different from what we have had previously, I'll be talking more on how we can prevent antimicrobial resistance using probiotic and prebiotics. And my presentation talks more about One Health approach. And as you listen, I will enjoy you to sit back. All right, can you see the screen?
Yes, I can see it. Go ahead.
All right. Sorry. Right. So I'll be talking on tackling antimicrobial resistance in poultry production using prebiotics, phytobiotics, and probiotics. We have all been aware of global challenge of antimicrobial resistance, and we've seen what havoc is before us due to antimicrobial resistance. And like we've all known that it has been predicted in 2050 that about 10 million persons may die annually if this is not being mitigated. So there's a connection between AMR, animal production, and public health, as I'll be discussing in the course of my presentation. But why I decided to talk on poultry industry is because the global population of poultry exceeds 23 billion, approximately 3 persons for each individual on planet, as stated by FAO statistics in 2016. So it is also stated by Alexandros and Brusima that world production of poultry meat is expected to increase by over 181 million tons by the year 2050. And of course, by this year, human population has been projected to hit 9.8 billion as stated by United Nations in 2017. So this means that there is an impending doom before us if something is not being done quickly regarding AMR. And, looking at the population of poultry and looking at how that as human population increases, as poultry population increases, human population, as it increases, we discovered that many people, especially in the rural setting and in low and middle income countries, we discovered that most of them, their source of income is actually poultry. And many farmers don't actually observe withdrawal period whenever their birds are being medicated in this region. So it was discovered by Klein et al. 2018 that the use of antimicrobial will witness a 65% increase from the year 2000 to 2015. And of course, looking at the current statistics, that should have been more than that. So the increasing rate of resistance to commonly used antibiotics has actually brought about serious issues that is likely to affect both animal production and human populace. So, and commonly used antibiotics in animal production include tetracycline, sulfonamide, and penicillin. So you could see that both humans and animals have a kind of shared use of antimicrobials. So from this graph, you could see that poultry meat is having a kind of sharp surge from 1990 to about 2028. And this is a projection. And you could see from other livestock meats, such as pig, beef, and sheep, you could see from this graph that poultry meat seems to have the highest. So, which means that poultry production is actually very important as far as human population is concerned. And looking at this flowchart, you discover that as income and urbanization increases, as you discover that there will be an expected increment in the quantity of meat that is going to be consumed. And like I stated earlier, that by 2050, about 181 million tons of poultry meat are to be consumed. And all these things has been attributed to the fact that there will be more income and there will be more urbanization, which means that many people will migrate from rural setting to urban setting, which actually posed a challenge and will eventually lead to increment in the use of antimicrobial substances. So, as population grows, there will be more demand for poultry products such as meat and egg, and there will be intensification of animal farming, and this will lead to antimicrobial usage in animal production, and all these things. Farmers actually use antimicrobial to for growth promotion and to also bring about disease prevention, but we discovered that especially in low, middle, and income countries, many farmers actually use antimicrobials indiscriminately, which actually bring about concern about antimicrobial resistance. And that is why in my talk today, I felt it necessary to talk about alternatives to antimicrobials, such as prebiotics, probiotics, organic acid, and phytobiotics. You also discover from this flowchart that as more antimicrobials are being used, then there is likelihood that we will be having resistant bacteria in animals, and this will actually bring about risk of zoonotic disease and eventually public health implication. So this flowchart is actually an interplay of poultry production, population growth, and antimicrobial resistance. So mode of AMR transmission in poultry production, we discovered that we have human exposure, consumption of contaminated food, and also water and soil contamination. Now, humans are being exposed in poultry farms, in slaughterhouses, in processing plants. They are being, these plants, they're actually being exposed whenever antimicrobials are being used indiscriminately in farms that are especially found in low and middle income countries. And as people get to eat meat and egg, they become further exposed to AMR. Then we also discovered that in farm settings, whenever antibiotics are being used indiscriminately, and if in farm produce or some of maybe like manure, whenever they're actually being excavated in the farm, and if these manure are containing resistant bacteria, and if they are being run off from farms, we discovered that it will affect human population. So we can see from this interplay, having resistant bacteria from poultry farms, slaughterhouses, and processing plants, and having from meat and egg, and from runoff farms, all these things, whenever they play together, they affect human population. So consequences of AMR in poultry production, one is that it's reduced treatment efficacy. So infection in poultry become harder to treat, leading to increased mortality rate and economic losses for farmers. Then there is also risk of zoonotic transmission. Resistant bacteria in poultry can potentially transfer to humans through direct contact, consumption of contaminated meat, or environmental pathway, and there will be increased health costs. Treating infection in both animals and humans become more challenging and expensive due to limited availability of effective antibiotics. And lastly, there will be impact on food safety. So contamination of poultry products such as meat and eggs with antimicrobial resistant bacteria poses a risk to consumers, affecting food safety. So what actually causes AMR in poultry production? One is overuse and misuse of antibiotics. Many farmers don't heed to the instruction of veterinarians whenever they are to give medications to their animals, especially in low and middle income countries. So, and of course, like I said earlier, that in poultry industry, antibiotics are often being used to prevent disease and also for growth promotion. So overuse and misuse of antibiotics contribute to development of resistance among bacteria, which will eventually be passed onto poultry products such as meat and egg, and whenever humans consume it.
5 minutes left. Sorry, 5 minutes left.
All right. So other points are suboptimal farming practices. Then we have incomplete treatment causes and environmental factors. And I also discovered from my years of experience that farmers, especially where I'm currently based, don't observe withdrawal periods whenever they are medicating their birds. And all these things will actually pose a risk to both the humans and the animals alike. So this diagram is also an interplay of poultry production, population growth, and antimicrobial resistance. It further buttresses the flowchart I explained earlier. You could see from these antimicrobial agents being given by poultry farmers to chickens. So we will see that both wildlife and other breeds of poultry and domestic animals are actually at risk of being affected by antimicrobial resistant gene in these antimicrobials. And we could see that from this spillover event, human population are likely to be affected. So that brings us to the use of prebiotics, probiotics, and phytobiotics. So with the basic difference between prebiotics and probiotics is the fact that in probiotics, live beneficial organisms such as bacteria and yeast are being given to animals such as poultry, while in prebiotics it serves as a kind of a substrate, and they are non-digestible fibers that nourish beneficial gut microbes. So their mode of action for probiotics is to actually competitively exclude harmful bacteria produced by bioactive substances such as bacteriocin, organic acid, and hydrogen peroxide, and to also modulate immune system and immune response of these animals. The prebiotics actually act by selectively support good bacterial growth and produce short fatty acid through fermentation. So example of these probiotic organisms are lactobacillus, bifidobacterium, enterococci, and this can be present either in cheese, yogurt. It can also be present in form of pills and supplements. And we see the whole essence of probiotics is to actually bring about a kind of balance in microbiome of the animal, so that the animal can effectively fight against pathogenic organisms. So on my slide are actually the benefits of probiotics, and phytobiotics in poultry production. We can see that in all these things that is being stated here, these are all beneficial effects that the animals can actually have whenever any of these agents are being consumed. So common source of probiotics are, as stated on the slide, we have those sourced from inulin and fructooligosaccharides. These are the sources. So we have those from galacto-oligosaccharides, and these are the sources as it is on the slide. We have those from resistant starch, we have those from dietary fiber, we have those from pectin, we have those from beta glucans, we have those from arabinoxylan and polyphenol. Then we also have the ones from beta leaf, which I'm currently actually working on. So those from phytobiotics as stated on the slide also, we have from thyme, turmeric, ginger, garlic, aloe vera, neem, green tea, and cereals extract. All these things are actually a natural source that can conveniently replace the use of antibiotics. So I also discovered that the use of this natural source of probiotics and prebiotics and also phytobiotics should be strongly encouraged, especially in low income, and in low middle and income countries. This is because they are cost effective, they are accessible, they are easy to process, they are nutrient rich, they are sustainable, they are culturally accepted, and they have a microbial diversity, and also have synergistic effects whenever they are being used. This was a previous work that we conducted here in Nigeria, and that was the use of supernatants of fermented maize over a period of 6 weeks. And you could see from this graph, it was a study period that took us about 6 weeks. You could see that we divided the beds into about 6 groups. Group A were given
1 minute more.
All right. Group A are the negative control. B were given positive. They were positive control, which were given commercially prepared probiotics. Group C, D, and E were given natural source of probiotics, that is supernatant or fermented maize in graded dosage. And those group that were given 10 mils per liter, which is this group, you will see that they have the highest weights. So which means that giving birds, that is poultry, such as chickens and other types of birds, natural source of probiotics, such as supernatant or fermented maize, will actually bring about positive effect in these birds, which will eventually help in replacing the use of antibiotics and eventually help in mitigating antimicrobial resistance. So this was mortality rate. We could see that even from here, group C, which were given the highest dose of this supernatant of fermented maize, actually had the lowest mortality rate. So our current work using bitter leaf extracts, as seen on this flow chart, I may actually not say much about it, but what I will say is that we are having promising results through the use of bitter leaf extracts to actually serve as an alternative to the use of antibiotics. So we can see the work from this flowchart involve preparation and translation of aqueous bitter leaf extracts, then broiler chicken experiment. And from this experiment, our aim is to assess growth parameters. And we also take blood sample from this chicken to harvest the serum and to check the serum biochemistry and immune response. And from this, we also harvest tissue from the liver and proventriculus to actually do gene expression analysis. And from same chicken, we harvested intestinal content such as jejunum, duodenum, and ileum, so that we can analyze the microbiota, including pathogenic and beneficial microbes. And currently, we are at this stage. And in our future research, we aim to do metagenomic analysis of these organisms. And we've also done histological analysis. And maybe in the course of time, we might have the results presented. And a special outcome from this study is to actually see that there will be a solution of a balanced gut health microbiota in broiler chicken, reducing the prevalence of.
Hello, can you just round up now?
Feed conversion and efficiency and overall health of broiler chicken. Then we also expect to have promotion of sustainable and responsible poultry production practices to combat AMR using a natural source. So these are my references for this presentation. Thank you for listening.
Okay, thank you very much. I think that was very insightful, and I think a lot of people can actually relate to it. I can, because you're talking about poultry beds, which I see almost every day in Nigeria. They are currently residing now, so it's actually something we can easily. And every one of us actually love chicken and eating poultry beds, so it's very, very relatable. Thank you very much. So, Margaret, can we have you now?
Yes, sure.
You can stop sharing, David. You can stop sharing, David.
Right.
Hello. I'm going to have my video on for the introduction, and then I will put it off just because of bandwidth. So good morning, good afternoon, good evening, depending on where you are. My name is Margaret Lubwama. I'm a lecturer and medical microbiologist with Margaret University College, and my research focuses on antimicrobial resistance in the immunocompromised host, that is in cancer patients. And also I'm into medical education, designing antimicrobial resistant courses for preclinical and clinical students in our college. So I'm going to give a talk on the importance of genomic surveillance of antimicrobial resistance. So I'd like to acknowledge my funders who made this work possible. I have nothing to disclose. And this is the outline of my talk. I will give a brief background on the burden of antimicrobial resistance. I will mention how the WHO is addressing the burden of antimicrobial resistance through its global action plan on antimicrobial resistance. And then I will define genomic surveillance and briefly highlight the WHO global genomic surveillance strategy. And lastly, I will highlight how we are using genomic surveillance at the Uganda Cancer Institute. So looking at the burden of antimicrobial resistance is slowly creating, if not created, a post-antibiotic era. Moreover, an era in which we have no new antibiotics being produced, and this leads to increased morbidity, mortality, and healthcare costs. So bacterial antimicrobial resistance was directly responsible for 1.27 million global deaths in 2019. And also antimicrobial resistance could result in $1 trillion additional healthcare costs by 2050. And therefore, this is really a huge burden that we are facing. So to address this burden, the WHO came up with the Global Action Plan on Antimicrobial Resistance, which has 5 objectives, including improving awareness and understanding of antimicrobial resistance through effective communication, education, and training, strengthening the knowledge and evidence base through surveillance and research, reducing the incidence of infection through effective sanitation, hygiene, and infection prevention measures, optimizing the use of antimicrobial medicines in human and animal health, and developing the economic case for sustainability investment, and that takes account of the needs of all countries and increasing investment in new medicines, diagnostic tools, vaccines, and of course, other interventions. So the WHO, and specifically looking at the objective of strengthening the knowledge and evidence base through surveillance and research. The goal really is to develop an antimicrobial resistance surveillance system for healthcare facilities and community locally and internationally. So incidence, prevalence, clinical patterns related to antimicrobial resistance develops and spreads, including how resistance circulates within and between humans and animals and through food, water, and environment is important. And also importantly, the ability to rapidly characterize newly emerged resistance, any underlying mechanisms. So having that in mind, this genomic surveillance, it was not enough to know that organisms are present and what phenotype, what their phenotypic characteristics looks like, to know how the organisms are changing, that is mutating and spreading in the community. So knowing this can help us stop the organism in its tracks. But also genomic surveillance is all about. It is analyzing their genetic similarities and differences. And why is genomic surveillance important? Because we can track the path of epidemic, that is determine the rate of being formed. We can characterize the organisms, determine the species, antimicrobial mechanisms, all the new mechanisms, look at the virulence and how the organisms are changing and how the changes, excuse me, may impact diagnostics and therapeutics. Recently developed a global genomic surveillance strategy for pathogens with pandemic and epidemic potential. And we know that antimicrobial resistance is the next big. I'm not going to get into much of the strategy. You can find it in their website, but it's important to highlight that the goal pathogens with pandemic and epidemic potential strengthened and scaled for quality, timely, and appropriate public health action within local to global surveillance, in access to tools for better geographic representation, strengthening the workforce to deliver at speed, scale, and quality, of course, through training, enhancing data sharing and utility from streamlined local to global public health decision-making and action, maximizing connectivity and architecture. And I'm not going to go into detail. You can read about it in the website, but a strategy to address antimicrobial resistance, especially after the lessons that we've learned from the COVID-19 pandemic. So as an example, looking at genomic surveillance of pathogens causing bacteremia at the Uganda Cancer Institute. And when I say bacteremia, I mean bloodstream infections that are caused by bacteria. So we determined the characteristic of Enterococcus species isolated from blood of patients with hematologic malignancies at the Uganda Cancer Institute between November 2017 and December 2021. And these Enterococcus species were Enterococcus faecium, and I will walk you through that. But briefly, just for the sake of knowledge, Enterococcus faecium is a gram-positive facultative, the gram-positive facultative anaerobic escape pathogens, which are a leading cause of hospital-acquired to antibiotics such as cephalosporins and have reduced susceptibility to the aminoglycosides. And efficient strains reported in infections belong to the sequence within the. Our first step was to look at the conventional biochemical methods. We identified the organisms presumptively as Enterococcus species, and we went on to perform the antimicrobial susceptibility test using the Capybara disk diffusion method. And from this, we're able to tell, importantly, that the Enterococcus species that we isolated at the Uganda Cancer Institute were multi-drug resistant enough. we still had questions. So, for example, what species was it in our isolates at the Uganda Cancer Institute? Are they the same as the ones that are being reported elsewhere in the world? What mechanisms are responsible for virulent factors present? Was there an outbreak? So to answer these questions, we needed to study the genome. And so we carried out whole genome sequencing to obtain genomic characterization. And so we were able to tell concerning our isolates. We were able to confirm the species, which was Enterococcus faecium. And also we confirmed that during that time period, we had 3 circulating sequence types, all belonging to the clonal complex 17 and other centers worldwide. We also observed that sequence type 761 was found in Enterococcus faecium, isolated from 3 patients, all of the same ward and within a week. And 2 Enterococcus species, two Enterococcus faecium one day from 2 patients, but in different wards. One of the patients was in the peds ward and the other was in the adult ward. And so this may point to spread of infection within the Cancer Institute. And so this information that we've got from this genomic characterization can then feed guideline at the Cancer Institute. We were also able to show the presence of antimicrobial resistance genes, virulence factors, most of which are associated with different plasmids. Importantly, M, which has been associated with many phenotypic vancomycin resistance among us, that we pay attention and continue with surveillance so that we can catch any vancomycin resistance that may come up, especially the VAN-A type, which has been associated with this plasmid, spread with this plasmid. Also, through genomic surveillance, we were able to correct.
I said 3 minutes more. Sorry.
Through genomic surveillance, we're able to correctly identify a rare organism, which is Ishola confusa, misidentified as vancomycin-resistant Enterococcus. And using the automated machine, it was also misidentified as Streptococcus porcinus. Studies have shown that this organism, which is intrinsically vancomycin resistant, is commonly misidentified and requires genomic sequences or sequencing to correctly identify it. And then we are still trying to characterize the organism. However, when we clustered it with other Ishola species, it clustered with if it is a new strain that we're dealing with. So what are the implications for clinical care? It's important to note that surveillance, including genomic surveillance, is a team effort. It includes healthcare team, nurses, medical officers, pharmacists, laboratory, but also the administration needs to be involved. Surveillance requires personnel funding, and this will require administrative buy-in and approval. Also importantly, the patients should be involved. In this case, what we do as professionals affects people or community that funding of the patients should be considered in the decisions that we make. And then finally, it's important to share data internationally and locally so as to engage with other stakeholders stakeholders. So at the Uganda, 2 infectious disease doctors and a microbiologist has developed into committees. And all this was possible because we had the data. We're able to not only form the antimicrobial stewardship and infection prevention and control committees, but also inform the committees through our surveillance data. And so what we had was a translation of infectious disease knowledge into, so thank you so much for listening.
Thank you so much, Margaret. That was awesome. Thank you so much for sharing your findings and your updates on your work. Thank you so much. And Megan, can you go now? Megan, are you available? Can you unmute yourself and present now?
Yes.
Good. Do you want to share your slides? Oh, she has left the meeting. Okay. So I think there's a question for when we're still waiting for Megan to join again. David, there's a question. What do you think is actually contributing to the issue with poultry? Because when you show us the graph, it seems and even as you explained that there's a lot of antimicrobial resistance in poultry, more than very high compared to other livestock products. So can you kind of give us some key causes for that?
All right. When I was presenting, I talked about much of these issues being found in low middle income countries, and livestock generally. You could see from the data that poultry meat seems to be the most consumed. That's poultry products, meat and egg, seems to be the most consumed. Sorry, can I continue? Like, she's back.
Yeah, continue. Just a minute. So Megan, hold on
right.
for a minute, Megan, and then we'll come back to you.
All right. So poultry meat, poultry products such as meat and egg seems to be the most consumed. Like in Nigeria, where I'm currently based, data has it that there are about 181 million poultry birds. That's chicken. So, and the implication here is that the more of this we have, the more risk of AMR. And the reason for this resistance is simply because farmers don't adhere to vet doctors' instructions. Most farmers buy drugs at their own expense, and they don't bother to consult a vet. They just give the medication anyhow they like, and even if it's stated on the sachet that the drugs have so and so withdrawal period, most of them don't actually observe that. And of course, once that happens, what do we expect? We should expect that there will be more risk to AMR.
Okay. Thank you.
Yes.
So Megan, you can go ahead now.
Question.
Yeah, thank you. That was good. Thank you. Megan, you can go ahead now.
Okay. So I'm going to speak on AMR in humanitarian crises. Hopefully you can all see this and it doesn't go away. And so I'm going to speak on AMR and humanitarian crises. I'm Megan Coffey. I'm speaking about my work with the International Rescue Committee. I'm also in faculty at NYU Grossman School of Medicine. I'm an infectious disease attending physician, and I also teach at Columbia University on outbreaks and humanitarian crises. And then I work also with TCI. So I wear a few different hats. But so to give a little explanation about IRC, it's an organization that works a great deal with people who are in humanitarian crises. So it's to help people whose lives and livelihoods are shattered by conflict and disaster and the climate crisis. And what I'm really going to focus on is people who are in humanitarian crises. This is not people who are in low resource settings, who have had a storm. This is a complex emergency with displaced populations, internally displaced and refugees. UNHCR defines this as a crisis in any country or region where there is considerable breakdown in authority. So you're really breaking down governance, and usually because of internal or external conflict, which requires an international response. It brings in a lot of different actors, making it a much more complex situation. And to be honest, this is a setting where we have seen antimicrobial resistance flourish again and again. The first war in which there were antimicrobials, or at least penicillin, was World War II. And that's when we first saw some of the emergence. We saw it during wars in the 20th century. We've seen it more recently in Ukraine, Syria, and Libya, where we have really high levels of resistance being selected by the continual use of the same protocolized medications, high rates of empiric use, and also trauma. But to kind of put it in picture form, I kind of like to think of AMR as this really multi-prong effect, in that we think a lot in terms of what happens in clinical settings, in terms of clinicians making diagnoses and prescribing, pharmacists, diagnostic testing. We think of vaccination rates. Vaccine rates often drop off in humanitarian crises. We think of the availability of water in health facilities. As we know, many facilities will not have running water in primary health clinics or hospitals in settings that are in more complex situations, but even more so when you're faced with a disaster, you don't necessarily have the PPE to be able to protect against transmission. This will all in the clinical setting lead to the amplification of AMR, but there's also going to be a lot more who are injured, who seek care on their own, different sources and supply chain of antimicrobials. And then we've heard already about poultry, livestock, having close contact with livestock and the use of antimicrobials. And then what is the situation in terms of sewage? Is the normal situation in terms of wash, in terms of being able to have water and sewage disrupted? Do you have runoff from different facilities? Do you have runoff from livestock? And just knowing all these other factors in terms of that influence AMR, whether it's factories, whether it's fisheries, whether it's rivers, waterways, they all will contribute in some way. And then just knowing that when you have a complex emergency, you're going to have displaced populations that are often quite crowded. I mean, I think in people's minds right now are the images you have from Gaza of people being displaced and pushed into certain settings where they're going to be much more high risk for having infectious diseases, whether it's respiratory infections between each other or waterborne diseases because they have a disruption in their wash facilities. You're going to have more outbreaks. And as I said, you can have more who are injured, trauma, who will need antibiotics to treat these. So there's a very kind of complex picture. And then recognizing AMR is going to always have a breakdown in governance. That's kind of when AMR is going to flourish in a situation where you have a breakdown in governance in this sort of humanitarian crisis. And the governance to protect a common good or antimicrobials is always going to be an issue. I always speak of it in terms of the tragedy of the common, that if one of us brings our cow out and eat the pasture, it's going to be okay. But if we all do that, we're going to quickly run out of this. And that's the definition of tragedy of the common, but very much happens when we don't have the governance in place to be able to protect antimicrobials. And we'd have a lot of overuse of antimicrobials. And so just stressing that AMR is a lot more than just prescriptions in clinics. It's about community control, clean water, sanitation, animal husbandry, food preparation. It can be about individual control. Are you seeking vaccination? Do you have hand washing? Are you self-prescribing antimicrobials? How are you preparing your food at home? And then, of course, health facility control. What is your PPE, water facility? Not necessarily under the control of the people who work in the hospital, but it's a health facility issue. Sterile procedures and crowding. And all of these factors will really drive infection transmission, but also AMR. And I always kind of just bring up that it's the infectious disease burden, the transmission that leads to AMR, and of course, AMR increases the infectious disease burden, and it becomes this horrible vicious cycle. And then just to reiterate a little bit more about AMR and crises, you're going to have more infectious disease outbreaks. Cholera, malaria, measles are quite common friends of crises, and we should be expecting to see a lot more climate-mediated events. Climate change will push more people into crises. You run out of water, you run out of resources, you face disasters that were unexpected. This can really push populations, displace populations. The issues about having more malnutrition, recognizing that in crises, you're not able to get enough food, leads to infectious disease risk, and also the empiric and targeted antibiotic use. For severe malnutrition on the protocol is to give antibiotics, and it's the same antibiotics. So you really run the risk of amplifying the same resistance patterns. And as I said before, disrupted vaccination programs really increase the rate of chance of infectious disease spread, trauma, crowding, less sanitation capacity. Crises over again and again strike children more than anyone else. Most humanitarian crises have occurred in populations where the average age is often 18 or under. This is kind of the pattern again and again, and children will often have more infections. And this, particularly when you're going to be more stressed without diagnostics available, will lead to more antibiotic use, especially when you have no safety net, and giving antibiotic for what may be a viral infection with maybe a bacterial infection seems the most prudent. You're going to have strained health facilities and just other priorities. Anytime I work on AMR and humanitarian crises, I really need to remember we need to have a light touch. There are so many other priorities and so many security concerns, and the risk of lives of those who work in health facilities, of the communities that are affected, of health facilities being targeted by violence, and just communities in general not having a safety net. And this will really disrupt supply chain of antimicrobials and supply, and then knowing that you're going to have a limited supply of antimicrobials. And then recognizing, I'm going to speak soon about Nigeria as well. I'm speaking about Boko Haram affected areas historically in the Northeast. But so bringing up that in Nigeria, we've really seen while other infectious diseases have become less common, fewer deaths, neonatal disorders have remained the same, and so now have risen to be a leading cause of death in 2019. And so keeping that in mind is that in many settings where we have humanitarian crises, and we have high rates of birth of children in health facilities, we're often using antibiotics that were targeted to very different settings, where historically ampicillin and gentamicin are used because the big killers of neonates historically was group B strep, E. coli, and listeria. And that was also based on studies that were done in North America and Europe, finding again and again that in many settings in Africa, Middle East, in Asia, that the leading causes of neonatal sepsis are Klebsiella, Staph aureus, and E. coli. Without having the lab facility in a crisis, really important that we recognize that we may be driving some of the resistance, and knowing that crises happen where we have the least amount of data, and we often are that the story of the man looking for his keys under the streetlight, when of course his key is somewhere entirely different. And so we really need to be able to better understand, kind of finding data, finding the answers where they are. This was a slide from before, but recognizing that neonatal sepsis, since birth is going to be a common risk factor for AMR, that if you're giving ampicillin and gentamicin, if you look at ampicillin, which is on the far left, this is a study that includes, it's a Barnard study. It includes Nigeria, but includes many other countries. But the resistance rate is just astronomical for the gram-negative bacteria for ampicillin. And then gentamicin, which is in the middle here, gives you only about 25% coverage. So it's less than a coin flip. It's really a 25% chance that the regimen that you're giving this child is going to be working if they have a gram-negative neonatal sepsis. And we brought up before the outbreaks. very young populations. Again, empiric antibiotics are given for measles, severe malaria, severe cholera, malnutrition, even though on face value, most of these disorders would not require antibiotics. But in outbreaks and protocolized medicine, where we have fewer safety nets, we really do need to use antibiotics, but we may be losing these. And then saying before that AMR, social mortality, is probably highest in areas that are more affected by crises, and West Africa and Nigeria Nigeria is in the region, West Africa, that probably has the highest rate of mortality associated with AMR. But again, we don't necessarily have the data to be able to picture all of this. So what I'm going to talk about briefly, and I'd really like to be able to get some thoughts on kind of next steps, if I can ask that at the end. But looking at northeastern Nigeria, IRC has worked in Borno and Adamawa, which were areas that had been severely affected by Boko Haram. In the height of in 2016, there had been 6 million people whose lives were disrupted in Borno by Boko Haram, high rates of severe malnutrition, high rates of displacement, 2 million. So we looked at health facilities, and this is more recently in areas that have been rebuilding, in Maiduguri, the capital and the state in the center, and then some areas in the south. IRC also works in Adamawa, and it's hoping to expand there. And so this study we looked at from 2021 to 2023, it was funded by Pfizer, had 12 health facilities and associated communities, and did surveys for knowledge, attitude, and practice among the community members, understand what is the understanding of disease transmission, of antimicrobials, what are your practices and how do you obtain these, and how do you learn more? Who do you reach out to? Who do you trust? Stakeholders, what is your knowledge, your access to knowledge, your practice, your attitudes around antimicrobials and the concerns about AMR, and then quality of care assessments. Even if healthcare workers know all the correct protocols, it doesn't mean that they're able to wear gloves if there are no gloves, wash their hands if there is no water. But in general, there's often many hurdles that can affect how health practitioners will work, and trying to understand what are these factors. And then in between, there were going to be three CAP surveys done: first a baseline, a midline, and an end line. And there was education, SPCC, and an online course as well with the Nigerian CDC, running as well the curriculum that we worked with them to produce in health facilities and primary health facilities. We also worked with the University of Maiduguri to increase the microbiology capacity, BACTEC and VITEC approaches, and building up surveillance system bit by bit from primary health clinics, building partnerships, technical working group. This is my favorite part, is really bringing in veterinary leaders, pharmacy leaders, regulatory, water, sanitation, infection control, nursing, and really do gap analyses and understand. And then more SPCC with community outreach, meetings and radio and leaders. And then working more on partnerships, building up AMR committees, more of the surveillance, and actually designing the technical working group designed a state action plan, which was actually the first designed. There's a national action plan, but the state action plan for Borno. And this is the national action plan, which much of this work is based on, and protocols, again, much is based on. But trying to bring this all together and ask kind of, what I'd like to ask in the end is kind of what questions that the audience would really like to ask community members to better understand AMR in crises. This is kind of a resolving crisis, but we're going to say at the end, we're kind of moving into more active crises. In this study, I'm going to go very quickly through our results. I could go through 60 slides, and I'm not going to do that to you, but I'll just be very quick. The population we recruited at clinics, most were women, because most community members who come to clinics are women, either for their own care, for prenatal, postnatal, or for their children that they're bringing. And then also clinicians, most clinicians worldwide are also women. Here, most of the clinicians were clinical officers and then midwives and nurses. Most of the community members were mothers, and about half most had a child under 5, that's supposed to say, and over half had 4 children or more. About half had attended school, a quarter with primary education, and a quarter with secondary education. And about 3/4 were local population, and 1/4 was displaced persons from other parts of Borno. And just recognizing a high use of antibiotics. This survey was done in a clinic, but 4 out of 5 had used antibiotics in the last month. 5% of people reported their youngest child had never had antibiotics, meaning 95% of youngest children had antibiotics. About half will obtain antibiotics or antimillinars without going to the clinic. 3 out of 4, if they do go to the clinic and they're told they won't be given antibiotics that they feel they need, will go elsewhere. And about 1/3 have livestock or poultry around the home. And about half of these homes will give antibiotics to the poultry. Most people, there was the most common indication for using antibiotics here was colds. Most clinicians will prescribe antibiotics for URIs or COVID. Only 7% said they wouldn't give a healthy young man who came in with mild COVID antibiotic, if known to be COVID. Most clinicians did not recognize that neonates have AMR initially. And I said that we did a baseline and a midline. That was something that went up. They're only 31% knew initially, then 71% and more so recognize that neonates can have resistance, because that's a real concern, as I was stressing. And then a lot of extra use of antimicrobials beyond protocol, giving an extra antibiotic when only 1 was needed, giving a longer course. These were often issues where an antimicrobial was indicated, but less is more. And then supply chain, I always find this really fascinating. And I know this as a clinician, that past supply chain interruptions will affect your future use. If you have run out of RDTs, rapid diagnostic tests for malaria, again and again, you'll kind of save them. So even when you have them in stock, you're less likely to use them. We saw this in clinics that had a pattern of stockouts. Likewise, Flagyl or metronidazole was the most common antibiotic available, and then the most commonly used. Even when other antibiotics, which were the correct one for that indication, clinicians would often rely on what is constantly available and what they're more comfortable with. Clinical practice, and I can say this very much as a clinician myself, we often base on what we're most comfortable with when we're treating people. And then most clinicians said they would giving extra antibiotics probably causes no harm, and most thought that antibiotics would treat viruses. So just as a last thing, our work is extending now to Yemen and South Sudan, more active areas of humanitarian crises. We're going to be doing the baseline surveys and the quality control, the same kind of issues. And I'd be really interested in if there's any time, I mean, obviously from this, center and reach communities is a primary priority. Engage the primary health world in terms of AMR in crises. We really need a lot more advocacy. Humanitarian crises are very different than other settings. Best practices, pulling together lots of actors, the realities of the country, and then all the different organizations that come in crises, which can make a much bigger mess, so to speak, when you have all these different groups involved. And then recognizing protocols do not alone determine antimicrobial use. You may have maybe it's very difficult when you do not have diagnostics to know which antibiotic to give, what is the actual diagnosis. A lot will be based on the clinician's own assessment. And then recognize we need new metrics, new research, and more funding in humanitarian crises. So that's really it. Sorry for talking all that time, but if anyone has time, any questions you have for community members in AMR humanitarian crises, I would love to hear.
Okay,
Over.
thank you. Thank you very much for that. That was really exhaustive, and it was nice to really see what is actually happening, outside the everyday life, because we don't see these kind of things often. Well, context of humanitarian services. But can you kind of retrace the key drivers? That's like a question we have. Key drivers driving AMR prevalence in these settings, like just the key drivers.
Yeah, I mean, one I'm going to say is that we have young populations, we have crowding, we have lack of water, sanitation resources, and then we are going to not have the same ability to test and monitor. So we have to rely on a lot of antibiotics when we don't have a safety net. It's going to be a common response. Either personally, families are going to obtain antibiotics, or clinicians are going to give antibiotics when you don't have the ability to test and to have a safety net in a hospital to go to.
Okay, so thank you very much. That was nice. So it was nice to hear. To be honest, that was really nice. The storyline was just connecting. Okay, so thank you very much, everyone. That will be all from us. You can stop your video. Yeah. So and this is from now. So we have, let me just share my screen. We have one more webinar left, which will be next week, Wednesday. So you want to join us, and let's wrap this up together. I'm trying to share my slide. So yeah, so we have next week, we have Adela from I think from Netherlands. I think she's from Netherlands. Then we have Olawale, who is from Nigeria. Then we have Joseph Elikem, I think from Ghana. So that will also be very interesting next week. So join us again next week, Wednesday. If you've registered, you will receive an invite. And also the video of all our sessions will be on YouTube very soon. So thank you very much, everyone. That was nice from Megan, Margaret, and David. That will be all from now. And on behalf of Cecilia and myself, on behalf of Cecilia and myself, I want to appreciate everyone for joining. Thank you so much. And I feel like we had so much to discuss today, so much to digest about humanitarian crises, about poultry, and a lot of insight really. A lot of insight really to actually just think about even genomic surveillance from Margaret also. So that will be all, everyone, and bye for now.
All right, thank you.
Thank you. Thank you.